Hello, my name is George Tsokos, and I'm a rheumatologist at the Beth Israel Deaconess Medical Center and Harvard Medical School in Boston. My laboratory has been studying T-cell abnormalities in patients with systemic autoimmunity and mostly in patients with systemic lupus erythematosus. We've been impressed over the years with the T-cell that is unable to produce interleukin-2 while it produces excessive amounts of IL-17. In the course of our studies, we came across a calmodulin kinase 4, a kinase that phosphorylates serines and threonines, and we found that to account for the T-cell abnormality, and in parallel we found it to affect the function of resident kidney cells. Specifically, we found that CaMK4 through cyclic MP response element modulator alpha, CREM alpha, closes down the interleukin-2 locus and shuts off its production, while on the other hand, and simultaneously, it opens the locus of IL-17. Specifically, CaMK4 closes the locus by phosphorylating CREM and closing down the locus and simultaneously activating AKT mTOR pathway and increasing the binding of rho gamma T, the canonical transcription factor for IL-17 transcription. In the course of our studies, we made this mouse, that is the MRL-LPR mouse that develops spontaneous systemic lupus erythematosus, and we inserted the CaMK4 null locus. This mouse taught us that CaMK4 is important for both messenger cells, which it helps to proliferate, and podocytes, where it destroys their function. On the left side, you see a normal podocyte with a nucleus and its feet touching the membrane. The right side, I have a diagram indicating that podocytes that have increased amounts of CaMK4 display decreased amounts of nephrin, a protein that's absolutely necessary for the good function of the split diaphragm. Simultaneously, podocytes that express lots of CaMK4 overexpress on the surface the molecules CD80 and CD86, which have been described as stress molecules for podocytes, but simultaneously are known by immunologists to provide co-stimulation to T cells. When CaMK4 decreases, nephrin levels increase and the slit diaphragm functions properly, while the levels of CD80 and CD86 decrease significantly. We went into specimens from patients who had undergone kidney biopsy, and we first noticed that podocytes from patients with lupus nephritis expressed increased amounts of CaMK4. You can see that CaMK4 being in the areas where nephrin is also present, nephrin being a marker for podocytes. To our surprise, we found that specimens from patients with FSGS, that is focal segmental glomerulosclerosis, also express increased amounts of CaMK4, and that again in places where nephrin is also colocalized. We obviously went to mice to extend our studies, and we first noticed that mice that spontaneously develop lupus, that is the MRL-LPR mouse, express increased amounts of CaMK4 in their podocytes. We also look at mice that had been injected with adriamycin, and nephrologists have been using this to study in detail mechanistic aspects of FSGS. Mice that had been injected with adriamycin increased the expression of CaMK4 in podocytes. The same happened into mice that were injected with lipopolysaccharide, known to destroy podocytes or compromise their function. Then, at that point, we asked whether we could restore podocyte function if we could inhibit CaMK4 in a specific manner. For this, we teamed up with Dr. Tarek Fahmy of the Department of Bioengineering at Yale University. Tarek had been constructing nanolipid gels in which he packaged a CaMK4 inhibitor known as K93, On the outer surface of nanolipid gel, he placed streptavidin so that we can use any antibody to deliver the cargo to any cell we wish. In this case, we would use antinephrin or antipodocyte antibodies to carry the cargo specifically to podocytes. You can see in the lower part lupus mice that were treated with antinephrin-tagged K93-loaded nanolipid gels. Proteinuria disappeared completely. When we looked at the kidney pathology from these mice, you can see that glomerular lesions disappeared. And also please note that the crescents that are present on the left side have disappeared in the specimen on the right side that was from a mouse that had been treated with nanolipid gels tagged with antinephrin and loaded with K93. Then we moved on to look whether immune complexes had been deposited in the kidneys of mice that had been treated with nanolipid gels loaded with K93. As you can see on the right side, podocytes from mice that had been treated with antinephrin nanolipid gels loaded with K93 maintain absolutely normal function, and the podocytes display normal fit without effacement, and you see on the left side huge dense deposits that are usually found in kidney biopsies from mice that develop lupus in patients with lupus nephritis. This made us think that the immune complexes cannot deposit in the kidneys if the podocyte function is kept intact. Mind you that this mouse in the periphery, in the blood and the spleen and other lymph nodes, autoimmunity was rampant, but immune complexes were not deposited in the glomeruli. Here we administered nanolipid gels coated with antipodocyte or antinephrin and loaded with K93 to mice that had been exposed to adriamycin. Starting from the upper left side, you can see proteinuria is suppressed significantly, and on the right side you can see that the expression of CaMK4 decreases significantly, and the lower right side you see that the podocytes keep their structure and there's no podocyte effacement. Interestingly, when we gave to these mice nanolipid gels loaded with K93, 7 days after the injection of adriamycin, the process was completely reversed, giving us the hope that this kind of an approach can reverse established treatment. Subsequently, we performed biochemistry experiments to understand how CaMK4 compromises the structure and function of podocytes. Already mentioned that LPS increases the levels of CaMK4, and so does immunoglobulin from mice and patients with lupus, which enters podocytes using the FcRn receptor. When CaMK4 is increased, we found that it phosphorylates the scaffold protein 1433 beta, and when that happens, it releases synaptopodin, which subsequently is degraded. For CaMK4 to function, it needs well-functioning calcium channels, and if the calcium channels are inhibited, CaMK4 activity, of course, is suppressed. From the detailed experiments, I will show you only two pieces. Here you see podocytes in culture which display stress fiber on the left side. And on the middle side, you see how this is compromised when the podocytes are exposed to lipopolysaccharide. On the right side, you see that lipopolysaccharide cannot destroy the appearance of stress fibers if CaMK4 has either been inhibited with a small drug, the K93, or silenced with siRNA. I will share also with you, which we consider an interesting experiment. We plated podocytes to confluence, and then we made a scratch with a blade. As it can be seen, When CaMK4 was inhibited, cells did not move to fill up the wound. To the extent podocytes are the cellular source of crescents, this observation explains why delivery of the CaMK4 inhibitor to podocytes in lupus mice prevented the formation of crescents. Our studies have delivered two novel messages. One, which we consider important, is that CaMK4 is a shared molecule for the expression of podocytopathy in immune diseases and non-immune diseases. The second important point is that if someone is able to preserve the structure and the function of podocytes through the inhibition of CaMK4, this would not allow the deposition of immune complexes in the glomerulus, and this will reverse the classic dogma that immune complexes instigate inflammation in the glomeruli. Mind you that the inhibition that we accomplish here requires 10% or even less of the dose of CaMK4 inhibitor that would be otherwise needed if it were to be administered systemically. This study has enormous translational value because targeted delivery of CaMK4 inhibitor to podocytes should help patients with systemic lupus erythematosus and patients with focal segmental glomerulosclerosis. Before I end, I want to give credit to the people who are responsible for most of the work. Keiko Maeda performed the bulk of the experiments. Dr. Tarek Fahmy of Yale, with his associates, constructed the nanolipid gels and which we used in this study. Kotaro Otomo, Nobu Yoshida, Andrew Ferretti, Michi Kono, Ria Bhargava, in my laboratory contributed to different aspects of the work. Doctors Ikinose, Nishito, Mariyama helped us with patient samples. Dr. Abu Assad of the National Eye Institute helped with the EM data, and Maria Tsokos of Beth Israel Deaconess and Harvard Medical School provided conceptual help and also helped with the analysis of the pathology data. The study was supported by a grant from the National Institute of Arthritis and Musculoskeletal Diseases and the Lupus Insight Award. Thank you.